Hello, my name is Ayoob Redi and my presentation is on the topic, does pH level affect enzyme activity? Firstly, we must define what enzymes are. So basically, enzymes are proteins that speed up chemical reactions in our bodies by reducing the activation energy it takes to complete a reaction. Enzymes are important to living things because they aid in digestion, breathing, reproduction, and many other activities in our body. The enzyme that I will be using in my experiment is trypsin. Trypsin is a proteolytic enzyme made in the pancreas that is then converted into active trypsin in the lumen of the small intestine. Its function is to digest proteins. And for substrate, I'm going to use azocasein, which is a chemically modified protein and will be used in my experiment as the substrate for the enzyme trypsin. Finally, before discussing my hypothesis, I need to talk about pH level. pH level is a measure of how acidic or basic a solution is. It's ranged from pH 1, which is most acidic, to pH 14, which is most basic. So coming to my hypothesis, trypsin functions best at an optimum pH, a specific pH. Anything too acidic or too basic may negatively affect the enzyme activity. So my prediction is pH level between pH 7 and 9 is the best for the enzymatic activity of trypsin. The reasoning behind this is that trypsin is found in the pancreas. However, active trypsin is made in the small intestine, and the pH of the small intestine is approximately 8. So variables. My experimental variable was pH value pH 5 to pH 11, while my control variables were salt concentration and temperature. The chemical basis of the enzyme assay is that azocasein, an orange protein, is broken down by trypsin, an enzyme. Once TCA is added and the tube is pelleted, what is left is the supernatant. The more orange the supernatant is, the more active trypsin was during the reaction. For my experimental design, we took 500 microliters of azocasein and added it into 7 clean centrifuge tubes and labeled them and kept them on ice. Reaction buffers between pH 5 to 11 were added into each centrifuge tube. Then after that, 550 microliters of trypsin was added into those 7 tubes. Each tube was incubated for 10 minutes. Then 500 microliters of 10% TCA was added to each tube and mixed. Each tube was centrifuged for 2 minutes, and then the supernatant was transferred into a clean cuvette. A spectrophotometer was set to blank at 440 nanometers using a cuvette with DH2O. The absorbance was recorded in the spectrophotometer, and then the experiment was repeated 3 more times for more accurate data. At the end, the experiment was repeated 1 more time, but this time to find the negative control by replacing the 50 microliters of trypsin with 50 microliters of deionized water to get a negative control, which means seeing what the azocasein would do without the trypsin. At the end, we were left with 4 values and 1 negative value for each pH level from 5 to 11. As you can see in the results, we got 4 values for each pH and 1 for each negative control, and using the values along with the standard deviation, we were able to make a bar graph. And as you can see right here, the highest enzymatic activity is around 9, but the deviation overlaps between 8 to 10. Also, we had to conduct a t-test in which if the p-value was less than 0.05, then there was no significant difference, and if it was higher than 0.05, it meant that there was a significant difference for each pH between 5 to 11. And we found that, as you can see right here, that pH 8 and 9 were not significantly different, and pH 9 to 10 were not significantly different. And the conclusion of the t-test was that trypsin's optimal enzymatic activity range is between pH 8 to 10. So in conclusion, according to the t-test, our hypothesis has been disproven as it states that the pH range for enzymatic activity of trypsin is 8 to 10. However, our hypothesis was that it was between 7 and 9, which is why it's wrong. Also, the deviation of pH in the bar graph overlaps between 8 and 10, and that's an additional reason why. Discussion. I believe that the design and execution of my experiment was valid due to the fact that we could figure out the range of which pH was best for the enzyme trypsin. The relevance of my conclusion to another real-life example is that just like trypsin, other enzymes need a specific pH to operate at. For example, the enzyme pepsin operates at a range between 1 to 2 pH, and if met with a pH of anything higher than 3, pepsin will become completely inactivated, which means that pepsin only works in highly acidic solutions. Potential source of variability I missed out on in my experiment was that I had to test multiple times for the negative control, but instead I tested once, which means that my negative control isn't that accurate when compared to my other results. I don't believe that my control variable resulted in the highest possible rate of enzyme activity because there are many other factors such as salt and temperature that could also affect enzymatic activity of trypsin, and I have not tested for them yet. What I would do differently in the future is increase the variability of my results by testing a lot more times for the negative control and perhaps even incrementing at a value less than 0.1 for each pH, meaning that I would go from pH 8 to 8.1 to 8.2 to 8.3 to figure out what pH exactly is best for trypsin. My references are Suji and Deniston from Department of Biological Sciences at Towson University, and also an article called Pepsin, an Overview by scientificdirecttopics.com. Thanks for listening.